Welcome to this 19th lecture in this course on eukaryotic gene expression, basics and benefits. Today we are going to discuss about regulation of gene expression by growth factors. Just to recapitulate what we have been doing in the last few classes is, since last 4 classes we are primarily discussing about signal transduction pathways and regulation of gene expression. The focus is primarily to see how gene expression inside the nucleus is modulated by molecules which either enter the cell through the cell membrane or molecules which stay at the cell membrane. And what we have been discussing in the last few classes is that molecules such as the lipophilic signaling molecules, they can simply diffuse through the cell membrane and interact with specific cells intracellular receptors, and these receptors can then go on and bind to specific sequences in the promoters of target genes and either activate or repress transcription. We still have not discussed in detail this particular pathway, but what we are actually discussing in the last few classes is about another set of signaling molecules which are rather water soluble, and therefore they cannot pass through the lipid bilayer. Therefore they stay at the cell membrane and interact with specific membrane receptors, and when these molecules interact with membrane receptors, that results in what is called as a signal transduction events, ultimately leading to the activation of specific protein kinases, and these kinases then go inside the nucleus, phosphorylate certain transcription factors, which in turn either activate or repress transcription of specific genes, leading to specific physiological responses. Now, signaling by membrane receptors is a huge area of research. A number of signaling molecules activate gene expression through this pathway. We actually began discussing one set of molecules which interact with specific receptors called as the G protein coupled receptors or GPCRs, which are 7 transmembrane proteins. And what we discussed is when molecules like epinephrine or glucagon, when they interact with such 7 membrane GPCRs, it then results in the activation of trimeric G proteins, which consists of alpha, beta and gamma subunits, and then the alpha subunit dissociates from the beta gamma subunit. The alpha subunit then goes and activates molecules such as protein kinase, phospholipase C, or it can activate protein kinase A, or it can activate protein kinase C, and depending upon what kind of G alpha subunits are interacting with this, you have specific second messengers being activated. For example, when G alpha subunit interacts with protein kinase A, it results in the synthesis of cyclic AMP. When the G alpha subunit interacts with phospholipase C, it results in the production of calcium and diacylglycerol, which then go on, then all these act as second messengers. And we also discussed about guanylate cyclases, which unlike the other ones is an intrinsic membrane receptor having a guanylate cyclase activity, and that also gets activated by specific G protein coupled receptors, and that results in the synthesis of cyclic GMP, again acts as a very important signaling molecules. So we basically covered one particular group of signaling molecules which activate gene expression programs through GPCRs or G protein coupled receptors. What we will do in this lecture is to discuss another class of signaling molecules which activate gene expression programs through receptors known as receptor tyrosine kinases or RTKs. These are the receptors which have intrinsic tyrosine kinase activity. Therefore, when these signaling molecules bind to the receptor, the tyrosine kinase activity of the receptor is activated, and let us see how activation of this tyrosine kinase activity can pass through signals ultimately into the cell, leading to the activation or repression of transcription. Now, this kind of signaling pathway that we are going to discuss today is primarily mediated by molecules known as growth factors. So, let us now try to understand what are these growth factors, what kind of growth factors are influence gene expression programs through this particular pathway involving the receptor tyrosine kinases. Now, growth factors play a very important role in promoting cell proliferation, and we all know cells actually cycle, and this is what is known as cell cycle. And when cells cycle, they usually enter from gap 1 phase, when cells actually grow, increase in size, and make sure all the raw materials for the cells division of replication of DNA as well as other processes are present. And then from the G1 phase, cells enter the S phase, then go to the G2 phase, then enter the mitosis. This is what is called as cell cycles. So growth factors primarily activate the cell cycle program and make cells commit from G1 to and enter the cell cycle, and this is what thus induce the cell division, leading to cell proliferation. So what we will now discuss in the next few minutes is to understand how these growth factors induce cell cycle and make cells to commit from G1 to S, and how the cell cycle progression is initiated by growth factors leading to cell proliferation and cell growth. Now, let us go to a brief history of how growth factors were actually discovered, because it is very important for us to understand, appreciate how the field has grown over the last few years, ever since the discovery of growth factors by two very important pioneers in this field, Stanley Cohen and Rita Levi-Montalcini. These are the two people to whom the credit for discovering growth factors goes, and to acknowledge their contribution to the field of science, to the field of biology and medicine, Nobel Prize was given to them in the year 1986. So, as you can see, the citation in the Nobel Prize website says the Nobel Prize for Physiology and Medicine 96 was awarded jointly to Stanley Cohen and Rita Levi-Montalcini for their discoveries of growth factors. So, growth factors are very important, and the discovery of growth factors has been acknowledged by awarding Nobel Prize to these two people. So, let us now see what exactly these people did and how exactly growth factors were discovered. Now, sometime in 1952, Rita demonstrated that tumors from mice, when transplanted to chick embryos, induced potent growth of chick embryo nervous system, specifically sensory and sympathetic nerves. So, when she took the tumors from mice and implanted chick embryos, it promoted the growth of nervous tissue in the chick embryo. But an important observation that she made is that this outgrowth did not require direct contact between the tumor and the chick embryo, clearly indicating that the tumor actually released a specific nerve growth promoting factor, which induced growth of nerves in the chick embryo. So, some kind of a diffusive molecule is coming out of these tumor cells, that is what is acting on this chick embryo tissue and then inducing the proliferation of or inducing the formation of nerve tissue. So, this work was going on around 1958. Stanley Cohen actually purified this factor, she called it as nerve growth factor, because it was actually promoting the growth of nerves. So, 1958, Cohen actually purified this nerve growth factor from the salivary glands of adult mice and snake venom. And he also made another important observation, that while actually studying this nerve growth factor from the salivary, purified from the salivary glands, he observed that the salivary gland extract contains another important growth factor, and he called, which is different from that of the nerve growth factor, and he called it as epidermal growth factor, or EGF, because it basically stimulates the proliferation of epithelial cells in the skin and cornea. So, this is how this marked the beginning of discovery of growth factor research. So, Cohen and Rita Levi-Montalcini are the two people who discovered two important growth factors, namely the nerve growth factor and the epidermal growth factor, and this initiated the growth factor research. And today we have a whole battery of growth factors, and these growth factors and growth factor receptors has now grown into a billion-dollar industry, because this growth factor signaling plays a very important role in areas like cancer, inflammation, and so on and so forth. So, a number of pharmaceutical companies are interested in developing molecules which either activate this growth factor signaling pathway or inhibit this growth factor signaling pathway, so that specific gene expression programs can be either activated or repressed, and that has very important biomedical applications. So, it is very important for us to understand how these growth factors, when they interact with these growth factor receptors, ultimately initiate a signal transduction pathway leading to activation of specific gene expression program, because it not only has implications in basic research, but also has a lot of importance in biotechnology. Now, the next question, once Cohen and Rita Montalcini discovered these two growth factors, the next question is, how do these growth factors act? How are they able to induce the growth of nerves, or how are they going to induce proliferation of the epithelial tissues? It turned out two important discoveries paved way for understanding this mechanism of action of growth factors. One is Tony Hunter and Bartholomew Sefton, working in Salk Institute in San Diego, discovered that proteins can be phosphorylated on tyrosine residues. Since then, phosphorylation of proteins on serine and threonine residues was known, but for the first time Tony Hunter actually showed there are protein kinases which can phosphorylate tyrosine residues, and this marked the another very important area of research called as discovery of receptor tyrosine kinases. The discovery of tyrosine kinases is another important landmark in the area of biology and medicine, because it again has very important implications in both biology as well as medicine. And once around the same time when these people discovered the tyrosine phosphorylation and tyrosine kinases, Cohen actually observed that the receptor for the epidermal growth factor in fact towards itself is a tyrosine kinase, and its activity can be induced by the binding of the ligand. This is the paper that was published by Cohen's group demonstrating that the receptor for the epidermal growth factor, or the EGF receptor or EGFR, is a tyrosine kinase, and when the growth factor binds the growth factor receptor, the tyrosine kinase activity of the receptor is activated. So these are the two important landmarks in the area of growth factor research, and this actually marked this opened up a entirely new area of research to understand how receptor tyrosine kinases play a very important role in number of cellular processes. So let us now try to understand how exactly the EGF signaling takes place, what happens when epidermal growth factor binds to the epidermal growth factor receptor. So I have now shown in this cartoon, it's actually taken from a very nice website. So I have just taken the slides from this website, which very nicely shows, for example, the epidermal growth factor receptor basically contains three important regions. One is the extracellular domain, to which the epidermal growth factor comes and binds, and it also contains a transmembrane domain. Now I would like to spend a couple of seconds here just to tell you that this transmembrane domain of the epidermal growth factor receptor is different from the G protein coupled receptors which we have studied in the last 3 classes. Now the GPCRs, if you recall, actually contains a 7 transmembrane domain. That means this transmembrane domain traverses the cell membrane 7 times. So these are called the serpentine receptors, or the 7 transmembrane receptors. Unlike that, the receptor tyrosine kinases contain a transmembrane domain which traverses the membrane only once. So it's called a single pass transmembrane domain. This is a very important distinction between the GPCRs, or the G protein coupled receptors, and the receptor tyrosine kinases. They contain serpentine or 7 transmembrane domain. This contains only 1 transmembrane domain. The third important domain that these receptors contain is as called the intracellular domain, and this intracellular domain, which actually faces the cytoplasm, is actually contains the tyrosine kinase activity. So the entire growth factor signaling hinges on two things: what happens when the growth factor binds the growth factor receptor, how the tyrosine kinase activity of this receptor is activated, and once the tyrosine kinase activity is activated, what happens next, how this tyrosine kinase now interacts with other proteins, and how this results in the phosphorylation or activation of other kinases, serine kinases, namely the MAP kinases, and which then go on and phosphorylate specific transcription factors, leading to activation or repression of specific target genes. So let us now try to understand how does the EGF receptor function in a normal cell. So this is a cartoon that tells you in a very nice manner, again taken from a very nice website. Here is a cartoon that actually depicts when the epidermal growth factor binds to the epidermal growth factor receptor, it induces dimerization of the receptor. I will just go back and then click once again. This is the epidermal growth factor, and you can see now when the epidermal growth factor binds to epidermal growth factor receptor, it now induces the dimerization of the receptor, and once the receptor dimer is formed, it can now bind ATP, and using the energy released from ATP hydrolysis, now these tyrosine kinases transphosphorylate each other. That means one monomer phosphorylates the tyrosine residues on the other monomer, and this is called as a transphosphorylation. So each monomers phosphorylate tyrosines on each other, and thus the tyrosine kinase gets activated, and then very important events takes place. I have shown here as a blocked red arrow, which we will discuss in detail, and this activation of the tyrosine kinase activity leads to a series of events, including activation of what are called as the MAP kinase cascade, ultimately culminating in the regulation of gene expression inside the nucleus, and depending upon what kind of genes are getting activated or repressed, you can get cell proliferation, you can get angiogenesis, you can get anti-apoptosis, and all these events have a very important implications in cancer cell proliferation and finding a cure for cancer. So epidermal growth factor as well as other growth factors are very important targets for people working in the area of cancer, inflammation, and other areas of research, because activation of this particular signaling pathway ultimately results in proliferation of cells, leading to growth and cell proliferation. So let us now try to understand this red arrow, what exactly happens once the growth factor bound to its receptor and the receptor got activated, how does it influence events in the nucleus. Now what basically happens is that a number of inactive intracellular signaling proteins which are actually in the inactive state, and once the tyrosine kinase is activated, they all get activated and each one of them activates the member in the subsequent lineage of this pathway, and this series of activation of a series of molecules basically is now referred to as the MAP kinase signaling pathway. MAP kinase refers to mitogen activated protein kinases. So, these growth factors act as mitogens because they induce mitosis and make cells proliferate, and therefore, this particular kinases which are activated by mitogens is referred to as the mitogen activated protein kinases, because these protein kinases are activated by mitogens binding at the cell surface receptors. So, when the growth factor binds to the growth factor receptor, the receptor undergoes dimerization, and this dimerization activates the tyrosine kinase, induces a conformational change in the receptor, leading to the activation of the tyrosine kinase activity of receptors, and once the tyrosine residues in the receptor is phosphorylated, it activates the MAP kinase signaling cascade, and this activation of this MAP kinase signaling cascade ultimately results in the phosphorylation of specific transcription factors, leading to activation of cell proliferation programs. So, this is a very briefly the nutshell by which the growth factors influence gene expression programs. Now, ever since the discovery of epidermal growth factor as well as the nerve growth factor, a number of such growth factors have been discovered, and more or less they all act more or less in a similar scheme which I have described so far. That means, they all bind to a receptor which is a tyrosine kinase, and the activity of tyrosine kinase is actually activated when the hormone binds or the growth factor binds to the receptor. It promotes receptor dimerization, leading to the activation of tyrosine kinase activity. Now, some of these growth factors have been listed here, but this is not an exhaustive list. There are many others. I only listed a few here. For example, you have a growth factor called platelet derived growth factor or PDGF, which promotes proliferation of connective tissue, glial and smooth muscle cells. Epidermal growth factor we already discussed, proliferation of epithelial cells. You have what is called as a transforming growth factor alpha, which plays a very important role in wound healing. You have fibroblast growth factor, promotes proliferation of many cells, it may inhibit some stem cells and induces mesoderm to form in in the early embryos, plays a very important role during development, embryonic development. Nerve growth factor again was one of the original growth factor discovered along with the epidermal growth factor, promotes the outgrowth of neurites and neural cell survival. Erythropoietin is another very important hormone, promotes proliferation differentiation of erythrocytes. The transforming growth factor beta plays again a very important role in a number of cellular processes including anti-inflammation. It suppresses cytokine production, class 2 MHCP expression, promotes wound healing, inhibits macrophage and lymphocyte proliferation, and you have insulin like growth factor 1, insulin like growth factor 2 again has very important, regulates very important physiological responses. So, ever since the discovery of epidermal growth factor and nerve growth factor, a number of such growth factors have been discovered and some of this has been discovered. So, let us now spend a few minutes try to understand how these growth factors activate gene expression program, and we will use epidermal growth factor as a model or to understand how this growth factor signaling actually takes place. So, in general many of these growth factors, not all, but many of these growth factors, they have their own distinct receptors. So, each growth factor binds to its cognate receptor, and one thing that is common for many of the growth factors that we are going to discuss today, they all contain intrinsic receptor tyrosine kinase activity. So, they are all tyrosine kinases. The tyrosine kinase activity is activated when this growth factor binds to its cognate receptor. Now, once the tyrosine kinase activity of the receptor is activated, what kind of genes are activated or what kind of signaling pathway is activated depends on what are the effector molecules that are activated by the tyrosine kinase. In some cases, the activation of the tyrosine kinase activity results in the activation of phospholipase C. In such case, a different kind of signaling cascade is activated. If this growth factor receptor is activated RAS protein, which is a monomeric G protein, then a different kind of a signaling cascade is activated. If you have PI3 kinase as the effector molecule, when that is activated, a different kind of signaling cascade is activated, leading to activation or repression of different kind of target genes. So, in all these cases, basically it involves the activation of what are known as the mitogen activated protein kinases, and these mitogen activated protein kinases are nothing but serine threonine kinases. We have discussed some of these things in the GPCR signaling in the previous classes, and these MAP kinases ultimately phosphorylate specific transcription factors, leading to their activation, and these transcription factors then activate many genes involved in growth, such as the EGR1, CFOS, etc., leading to cell proliferation and growth. So, this is the general scheme by which many of these growth factors promote cell proliferation and growth. Now, so what I am going to do in the next few is I am going to run a series of cartoons just to give you an idea what are the important biochemical events that actually takes place, so that you can easily remember the various important molecular event that takes place leading to the activation of growth factor signaling pathway, ultimately culminating in the activation and repression of genes. So, what we will do in the next few slides is to go through a series of schemes to understand how exactly the signal transduction pathway is activated. So, what the purpose of this presentation is, once the growth factor binds to the growth factor receptor, how does specific genes inside the nucleus are activated, what are the events between the growth factor binding to the growth factor receptor and activation of genes involved in cell proliferation. This is what we are going to discuss. So, the first thing that happens when the growth factor binds to the growth factor receptor is the two monomers which were apart, which were far apart, now they come together and they undergo dimerization. So, when the growth factor binds, the immediate consequence of the growth factor binding to the growth factor receptor is the dimerization of the growth factors. And you can see here an important effective molecule in the signaling transduction pathway is the RAS-GDP. Now, when we were discussing about the GPR signaling pathway, we had discussed primarily about trimeric G proteins which contain 3 subunits, namely the alpha, beta and gamma, and we showed the activation of this trimeric GTPase actually depends on the dissociation of the beta gamma subunit and the alpha subunit which is now active, which then goes and interacts with a number of effector molecules like adenylate cyclase, or it can be phospholipase C and so on and so forth. Here what we are discussing is about a different kind of a G protein which has only one subunit, and this is known as the RAS. So, RAS is a monomeric GTPase and in many ways it has very high degree of homology to the G alpha subunit of the trimeric G protein which we have studied in the previous classes. So, the purpose of the next few minutes is to understand how this RAS, which is actually a GTPase, converts GTP to GDP, and how this RAS is activated by this growth factor binding to the growth factor receptor. What are the important events that takes place between the activation of RAS GTPase and the growth factor binding. Now, once the growth factor binds to the growth factor receptor and the receptors undergo dimerization, the immediate thing that happens is the transphosphorylation. So, tyrosine residues on this particular monomer is phosphorylated by this kinase of this kinase monomer, and the tyrosine residues on this particular monomer is phosphorylated by this particular kinase. So, transphosphorylation of tyrosine residues occurs on the monomer, and this results in the activation of the receptor. Now, once the tyrosine residues in the cytosolic domain of this growth factor receptor is phosphorylated, the next immediate event happens is these tyrosine phosphorylated growth factor receptors now interacts with a protein called GRB2. Now, GRB2 interacts with this tyrosine phosphorylated growth factor receptor through a very specific domain known as the SH2 domain. Now, SH stands for Sark homology domain because this domain was originally identified in a protein called SRC or Sark, which is an oncoprotein. So, once the tyrosine residues of a growth factor is phosphorylated, these phosphorylated tyrosines now interact with a protein called GRB2 through this SH2 domain. This is very important for you to remember because this particular interaction of this GRB2 with phosphorylated tyrosine kinases happens through this SH2 domains in a number of growth factor signaling pathways. So, interaction of this GRB2 with tyrosine residues of receptor requires these SH2 domains. Now, what happens next? Once the GRB2 interacts with the through the SH2 domain with the phosphotyrosine, it now interacts with another protein called SOS through another domain now known as the SH3 domain or the Sark homology 3 domain. So, the SH2 domain facilitates interaction of the GRB2 with the receptor tyrosine kinase, whereas the SH3 domain facilitates interaction of the GRB2 with SOS. Now, SOS is another protein known as the son of 7-less. Now, we will not go into the details of this because we are not really discussing the signal transduction lectures, but our primary aim is to understand how gene expression programs are activated through the signaling pathways. Now, GRB2, once the GRB2 interacts with the receptor tyrosine kinase through the SH domain, it undergoes a conformational change, and now because of this conformational change it can now interact with SOS through this SH3 domain. Now SOS is a very important protein, and this is a GDP exchange protein. SY is now capable of exchanging GDP on RAS for GTP. So the RAS which is in a GDP bound form, now the GDP is removed and it now is made to bind GTP. So the regulation of RAS activity is primarily carried out by SOS, and let us now understand how exactly this takes place. You can see RAS molecule, which is a GDP, which is basically a GTPase, when it is bound to GDP, it is inactive. That is why I have shown it in the red. So the inactive form of RAS, when it interacts with SOS, the SOS now removes the GDP and now makes the RAS bind to GTP. Now being a GTPase, so RAS, the SOS is actually called as a GTP exchange protein. It removes the GDP and replaces it with GTP in RAS molecule. So now we have RAS which is in the GTP bound form, and now in the GTP bound form it is considered active. So I depicted in the green here, and once being a GTPase, it immediately hydrolyzes the GTP and becomes again a RAS GTP. So as long as the growth factor remains bound to the growth factor receptor, there is a continuous activation of this transphosphorylation of tyrosine receptor occur. The GRB2 interacts with the phosphorylate receptor, then SOS interacts, and SOS now continuously exchanges GDP to GTP, and this signaling continues as long as the growth factor remains bound to the receptor. Now once the RAS is activated, now the immediate consequence is that RAS now activates a protein called as RAF, which is actually a kinase, and this RAF now activates a series of kinases, and this is known as the MAP kinase cascade. So we will spend some time to understand how this MAP kinase cascade is activated by RAS. So hope this particular important events are clear. I will go through this once again. Once a growth factor binds to a growth factor receptor, the two monomers of the growth factors dimerize, and once they dimerize, the tyrosine kinase activity is activated, and each monomer transphosphorylates the tyrosines of the other. And once the tyrosine receptor of growth factor is phosphorylated, it interacts with the protein called GRB2 through specific domains known as the SH2 domains, and now the GRB2 interacts with another protein called the SOS, which stands for son of 7-less, through another important domain called the SH3 domain, and SOS is known as the GDP exchange protein. It exchanges GDP for GTP in the case of RAS, and once RAS is in the GTP bound form, it hydrolyzes, and with GTP hydrolysis now results in the activation of RAF, which is a kinase, and this now RAF now goes and activates other series of kinases or results in the activation of the MAP kinase cascade. So we will now come to the second phase of this activation pathway. We have now finished one pass. So growth factor binding to growth factor receptor results in the activation of RAS. Now RAS results in the activation of the MAP kinase pathway. Now how does the MAP kinase pathway gets activated? The first one is known as the MAP kinase, which being in inactivated form is activated by RAF and RAS GTP. Now it becomes active. Once it gets phosphorylated, it now becomes active. Now phosphorylated MAP kinase now activates the next in the series, namely the MAP kinase, whose hydroxyl group of the serine now gets phosphorylated. So we have got an activated MAP kinase, and this MAP kinase now goes inside the nucleus and phosphorylates a number of substrate proteins which could include transcription factors and so on and so forth. So activation of this MAP kinase pathway is a very important event in the receptor tyrosine kinase signaling or the growth factor signaling. So once through a series of phosphorylations, different MAP kinases are activated, and finally the last MAP kinase now goes and activates, phosphorylates specific transcription factors, activating, and these transcription factors now go and bind to specific target genes, resulting in cell proliferation program. So we have now discussed of the three important events in the growth factor signaling. We have discussed two events. One is the growth factor binding to the growth factor receptor, leading to the activation of the RAS protein, and once the RAS protein is activated, it now results in the activation of a MAP kinase. Now let us see what does this MAP kinase do. Now MAP kinase now goes inside the nucleus and phosphorylates protein factors, transcription factors like the serum response regulatory factor or the SRF. Again, we have discussed in detail the serum regulatory factor in the previous classes when we discussed about the GPCRs, how again cell proliferation pathways can also be activated by G protein signaling involving the GPCRs, and serum response factor is again a very important transcription factor which controls the expression of what is known as the immediate early genes. These are the genes which are immediately transcribed within a few minutes of addition of a serum to cells. For example, we know that for cells to proliferate, mammalian cells to proliferate in culture, you require serum, and serum basically contains the growth factors such as EGF, FGF and so on and so forth. So that is why cells to proliferate in vitro require serum, and serum through these growth factors ultimately activates the serum response factor, and this serum response factor now binds to what is called serum response elements in promoters of immediate early response genes such as CFAS, and now CFAS gene is now activated by RNA polymerase II, and the CFAS messenger pre-messenger RNA is now synthesized. This messenger RNA now goes into cytoplasm, gets translated into CFAS protein, and since CFAS protein alone cannot form a homodimer and cannot go and bind to the target sequence, it requires a heterodimeric partner. So the CFAS protein now combines with Jun, which to form a Jun-Fos heterodimer, and this Jun-Fos heterodimer goes and binds to what is known as the AP1 response elements TGA, CTCA of again the late response gene or growth promoting genes, and these Juns which are now synthesized now initiates cell division leading to cell proliferation and mitosis and so on and so forth. So this is basically the pathway by which a growth factor binding a growth factor receptor ultimately ends in the activation of genes involved in cell proliferation. Now, once these events that are taking place, the growth factor signaling also has to be switched off. So what happens in the normal cells, after a while the synthesis of the growth factor ceases, and therefore growth factor can no longer bind. Therefore the receptor cannot dimerize, the receptors become now monomers, and once the receptor become monomers, because the tyrosines are not phosphorylated anymore, GRB2 cannot interact with the unphosphorylated form of the receptor, and therefore it neither can interact through the SH2 domain with the unphosphorylated tyrosine kinase, nor it can interact with the SOS through the SH3 domain, and therefore the RAS GTP can no longer remains now in the GDP bound form because the SOS cannot exchange GDP to GTP. So RAS becomes inactive. Therefore the MAP kinase cascade, MAP kinase are not activated, and therefore these genes, the serum response factor which is now in the phosphorylated form is now dephosphorylated, and it can no longer be phosphorylated by the MAP kinases. Therefore the FOS gene is not transcribed, FOS protein is not made, and FOS protein has a FOS mRNA has a very short half life. So the messenger which has already been synthesized is degraded, the FOS protein is also rapidly degraded, and as a result there is no longer activation of these genes involved in cell division and cell proliferation. Therefore cell division stops. So in the normal cells, the levels of growth factors are very nicely maintained. Therefore the growth factors go and bind the growth factor receptor only when necessary for a brief period, triggering the activation of the growth factor signaling pathway leading to cell proliferation, and once the growth factor synthesis ceases and when the growth factors are rapidly degraded, the pathway is no longer activated, and therefore the growth factor receptors cannot induce the activation of these target genes, and therefore cell proliferation ceases. Now the problem happens in cancer cells is that when this regulation is disrupted at any of the stages. In cancer cells the regulation can happen either at the growth factor level, or it can happen at the activation of the tyrosine kinase level, or it can happen at the RAS activation, or it can happen at the transcription level. Any abnormal event happening any of stages of cell signal transaction results in the continuous activation of genes involved in cell proliferation, leading to uncontrolled cell growth and cancer. Now I have given a very simplified version by which the growth factors now activate the promote cell proliferation by activating genes such as CFOS, which are known as the immediate early response gene, resulting in the activation of cell proliferation. But this is a very simplified version of the view. The purpose is not to confuse you, but to understand the basic aspect by which growth factors regulate gene expression, or growth factors activate the growth factor signaling pathway is activated. Just to give an example, among the various growth factors which I had listed earlier, such as PDGF, FGF, TGF and so on and so forth, the epidermal growth factor itself consists of a number of members in the epidermal growth factor family. For example, the normal epidermal growth factors which are present in normal cells is known as the epidermal growth factor receptor is known as the ERB1 or HER1 or EGF receptor, which is a normally present normal cells. It can bind to the growth factor, and when the growth factor binds, it can form a homodimers and result in the activation of tyrosine kinase domain, and then leading to all the signal transductions that we discussed so far, ultimately adding to the activation repression of target genes. But there are also now several variants of this growth factor receptor. This normal growth factor receptor is bound by molecules such as EGF, TGF alpha, beta cellulin, HBEGF, amphiregulin, and so on and so forth. Now, there are also several other versions. For example, there is a variant of this epidermal growth factor receptor which is known as the ERB2 or HER2, and this is present in mostly cancer cells. Now, the ERB2 differs from the ERB1 in the sense that it does not bind any specific ligands, and it actually in many cancer cells it actually acts as a dimeric partner. So, the ERB1 instead of forming homodimers, it can actually form a heterodimer with ERB2, and when this happens, it can lead to cancer. There are also other members known as the ERB3 or HER3, for which molecules known as heregulins actually bind. Again, I will not go into the details because we are not really interested in discussing in detail about the signal transduction pathways. I am just giving a very examples to tell you how complicated these growth factor signaling pathways, and even within just one growth factor named EGF, there are so many variants. I imagine there are so many such growth factors and so many variants have been reported. And for example, there is another one called as ERB4 or H4, again a different set of growth factors known as NRG2, NRG3, heregulins, beta cellulin, they bind. Now, each of them bind to the receptors, and this results in different physiological responses because they activate different cascading pathways. So, there are several variations in the EGF downstream signaling pathway. I have just listed some of the things. For example, when the epidermal growth factor binds to the epidermal growth factor receptor, in the case of ERB1 or HER1 or EGF receptor, it actually results in the homodimerization of the receptor, and this results in the normal signaling pathway which we have discussed so far. Now, in case of cancer cells, what happens? The same EGF receptor can actually now heterodimerize with the ERB2, and when this happens, this heterodimerization can actually lead to cancer. So, in many of the cancers, the ERB1 instead of forming homodimers can actually form a heterodimer with the ERB2, and these heterodimers can trigger uncontrolled cell proliferation leading to cancer. Similarly, there are many other different pathways like the heregulins and others also activate the receptors, but we will not go into details. So, just remember, so variants of epidermal growth factor receptor which are expressed in different cell types depending upon forming different kinds of dimers can bring about variations of the signal transduction pathway which I have just discussed, leading to different kinds of physiological responses. So, in the case of tumor cells, what happens? The epidermal growth factor, when it binds to the epidermal growth factor receptor and undergoes receptor dimerization, it can continuously activate either the RAS pathway, or it can also activate the phosphatidylanosinase kinase, resulting in the activation of what is called the AKT pathway, ultimately resulting in transcription of genes involved in cell proliferation, leading to cell proliferation. It can also lead to resistance of cancer cells to chemotherapy, radiotherapy, and can promote angiogenesis. It can also lead to metastasis, and can also make cancer cell resistant for apoptosis. So, you can see the EGF receptor signal transduction pathway in tumor cells is kind of slightly different from what happens in normal cells, and this differential signaling pathway or the uncontrolled proliferation of this pathway can actually result in activation of multiple signaling pathways, leading to uncontrolled cell proliferation and cell growth. Now, I had indicated the point that I am going to discuss now, that I have also indicated earlier when we were discussing about GPCRs, that these MAP kinases or these effector molecules like phospholipase C, protein kinase A or protein kinase C, they need not be activated only by one pathway, but they can be activated by multiple pathways. So, in this cartoon I am going to just illustrate the same point, wherein the same epidermal growth factor receptors can also be activated through G protein signaling. Here is a GPCR, the 7-transmembrane protein, and when the ligand for this G protein now binds, binding of this ligand to the GPCR can also trigger an event result. For example, when it activates a phospholipase C, the phospholipase C now results in the cleaving of inositol bisphosphate into inositol triphosphate and protein kinase C, and the protein kinase C now gets activated by diacylglycerol, and the inositol triphosphate now results in the release of calcium. This calcium through a different pathway can go and phosphorylate the tyrosine residue of growth factor receptors, and this tyrosine phosphorylation can then activate RAS pathway leading to MAP kinase activation and transcription of the target genes. So, you can see the receptor tyrosine kinases can be activated not only by the growth factor binding to the growth factor receptor, they can also be cross activated, or there is a crosstalk with the G protein coupled receptor pathway also. So, molecules or signaling molecules which bind to the G protein coupled receptors can also activate the receptor tyrosine kinase through a different signaling mechanism. So, when this G protein gets activated and when this G alpha now activates phospholipase C can cleave phosphatidylcholine bisphosphate, resulting in the generation of diacylglycerol and calcium, and the diacylglycerol and inositol triphosphate. The inositol triphosphate now goes to endoplasmic reticulum, resulting in the release of calcium. This calcium can now activate PYK2, which in turn can activate SRC, and this SRC can now go and phosphorylate the tyrosine residues, and this phosphorylation tyrosine residues now can activate RAS pathway, leading to activation of gene expression. There are also other mechanisms by which, for example, proteins known as matrix metalloproteinases can also release growth factors, and this release of growth factors, now the growth factor can go and bind to the receptor, growth factor receptor, promoting their dimerization, resulting in the activation of the RAS pathway, leading to activation of transcription. So, you can see the receptor tyrosine kinases can be activated not only by the growth factors directly bind to the growth factor receptors, they can also be activated through the G protein coupled receptor pathway, or they can also be activated by matrix metalloproteinases because they sequester the growth factors, and when they release the growth factors, now the growth factor goes and binds and then activate. Another mechanism by which this tyrosine receptor tyrosine can be activated is by steroid hormones. The steroid hormones can actually diffuse directly through the plasma membrane, and when these hormones diffuse through the plasma membrane, they bind to intracellular steroid hormone receptors, and when this steroid hormone receptor ligand complex now can go and bind to the promoter region of molecules genes coding for ERB2, and now the ERB2 gene gets transcribed and then get translated into ERB protein. ERB protein can now diffuse out of the cell, again come out, bind to the epidermal, bind to the growth factor receptors, and then induce again RAS activation, leading to activation of pathway. So, you can see the signal transduction pathways are not static. It is not that G protein coupled receptors can activate only one particular pathway and receptor tyrosine kinase can be activated only by certain molecules. There is a lot of crosstalk among the signaling pathways, and a particular receptor or a particular signaling pathway can be activated through multiple channels, and I have given just one example here to show how a receptor tyrosine can be activated either by matrix metalloproteinases or by G protein coupled receptors, or directly by growth factors, or they can also be indirectly activated by steroid hormones. So, you can see that is why the entire signal transduction pathways are very complicated, and it is not as simple as I have shown in the schematics in the previous few slides. So, the effects of activation of GPCRs and the receptor tyrosine kinase is more complicated than a step-by-step cascades which we have illustrated so far. In the last 3 classes we have studied GPCR signaling separately, now we are discussing receptor tyrosine kinase signaling separately, but in the cell there is a lot of crosstalk going on. Simultaneously, activation of a GPR signaling pathway can also result in the activation of receptor tyrosine kinase pathway, or when a growth factor binds to a growth factor receptor, it can also result in the activation of a GPCR. So, there is a lot of crosstalk that is going on. So, stimulation of either GPCRs or receptor tyrosine kinase often leads to production of multiple second messengers, and both types of receptors promote or inhibit the production of many of the same second messengers. So, the same kind of second messenger like calcium or protein kinase A can also be activated by either of these pathways. Receptor tyrosine kinase can also promote a signal transduction pathway that eventually acts on the same target as the GPCRs. So, the same target gene can either be activated by either the GPS signaling pathway, or it can also activate by the receptor tyrosine kinase pathway. For example, CREB can be activated either by the GPS signaling, or CREB may be also activated by a particular growth factor, because the second messenger that is activated probably in this case is a protein kinase A. Cyclic AMP, if the cyclic AMP synthesis leads to is the second messenger that is synthesized by both these activations, it can phosphorylate protein kinase A, and protein kinase A can now act on the CREB, and same kind of target genes can be activated. So, the point I am going trying to make is that signal transduction pathways are pretty complex. It is not as straightforward as we have discussed so far. There is a lot of crosstalk and multiple signalings can take place in the same cell, and the one particular signaling molecule can activate multiple signaling pathways. The same cellular response may be induced by multiple signaling pathways by distinct mechanisms. Like I just mentioned here, the CREB transcription factors can be activated either through a GPCR signaling pathway, or it can be activated either by the receptor tyrosine kinase pathway or many other signaling pathways that we are going to discuss in the next few classes. Interaction of different signaling pathways permits fine tuning of number of cellular activities. So you can see the activation or repression of target genes can happen through multiple signaling pathways, and there is a lot of crosstalk between these signaling pathways. Now, the epidermal growth factor receptor that we have studied so far is one of the most well characterized membrane receptors. A lot of things are known about it. The crystal structure is known, what kind of protein amino acids are phosphorylated, what kind of residues are phosphorylated, everything is known. This cartoon just tells you the various tyrosine residues that are actually phosphorylated by, and what is the function of these tyrosine residues, and what are the target proteins which interact with specific phosphorylated tyrosines. For example, GRB2 through its SH2 domain, it actually interacts with tyrosine 1092 and tyrosine 1110. Whereas if the tyrosine 1016 of epidermal growth factor is phosphorylated, it now interacts with phospholipase C. Whereas if tyrosine 915 and tyrosine 944 is phosphorylated, it interacts with SRC. So you can see how complicated the whole scenario is. So depending upon what kind of tyrosine residues are phosphorylated, the effector molecules with which the receptor is interacting can be different. And accordingly, the signaling pathways are also pretty different. Now, why is this epidermal growth factor and growth factor receptor so important? Here I have just given one example to show how in different kinds of cancers like breast cancer, colon cancer, lung cancer, head and neck cancer, ovarian cancer, pancreatic cancer, in all this cancer you can see the epidermal growth factor receptors are all highly overexpressed. For example, breast cancer, but the expression rate of EGF receptor is 14% to 91% above that expressed in the normal cells. So the epidermal growth factor is expressed very high levels in many of these cancer cells, because basically the cancer cells want to proliferate continuously. Therefore, by overexpressing growth factor receptors, it can be continuously bound by the growth factors, and as a result the growth factor receptor pathway can be continuously activated, resulting in continuous activation of these genes like CFOS, leading to continuous cell proliferation. So I can see in a number of cancers the expression rate of the growth factor receptors are pretty high, and this is one of the reasons why these cancer cells proliferate uncontrollably. There are also a number of variants of epidermal growth factor receptor which have been detected in a number of cancer cells. For example, there is one particular variant of epidermal growth factor receptor which has been shown to be expressed in cancer cell, which does not have an extracellular domain. Therefore, it cannot bind to the growth factor as I showed, whereas the normal receptor can bind. And this variant, therefore, the tyrosine kinase remains constitutively active. Therefore, it continuously interacts with GRB2, continuously activating RAS, resulting in continuous activation of MAP kinase pathway and continuous activation of the genes involved in cell proliferation. And this variant also cannot dimerize, and therefore its activity cannot be regulated. And these variants, such as what we discussed here, is normally found in cancer cell but not in normal cells, and such cells which express this kind of EGF receptor variants are highly susceptible to become cancerous. This is the cartoon that actually tells you what I have just described here. This particular variant of 3 does not have an extracellular domain, therefore the epidermal growth factor cannot bind to the extracellular domain. The tyrosine kinase domain is constitutively active, therefore it can, using ATP, it can get transphosphorylated, it also does not undergo dimerization, therefore it gets continuously phosphorylated on tyrosine residues, and as a result the signal transduction pathway is continuously on, and therefore there is continuous cell proliferation, anti-apoptosis, metastasis, which are all the hallmarks of cancer cells. So a number of cancer cells express variants of these epidermal growth factor receptors which have, by certain mutations, have lost the ability to bind ligand, and therefore the tyrosine kinase domain is continuously activated, leading to uncontrolled cell proliferation because of activation of these genes involved in cell proliferation. So the proliferation of EGF receptor, you can see in the normal cell the level of expression of the EGF receptor on the cell surface is very finely regulated. You only find a few receptors on the cell surface, whereas in the case of cancer cells, because of certain specific mutations, you can see the cancer cells, there is either upregulation, the number of receptors itself has increased, or there are certain variants of the receptors have been made. For example, there are receptors which lack the extracellular domain, and therefore they cannot dimerize, and therefore they can be constitutively activated, and all these events ultimately lead to uncontrolled cell proliferation and cancer. So, a number of cancers, either overexpression of epidermal growth factor receptor or expression of variants of growth factor receptors is primarily responsible for a normal cell becoming a cancer cell. Because of this particular observation, that epidermal growth factor receptor, there is a very good correlation between the levels or variations of epidermal growth factor receptor and cancer. The epidermal growth factor receptor has become an excellent target for cancer. So, a number of approaches to kill cancer cells or to treat cancer primarily revolves around epidermal growth factor receptor, and I will just give you one or two examples. For example, just in the previous slides I had shown the non-small cell lung carcinoma, the epidermal growth factor receptor is highly expressed, is highly overexpressed. Therefore, in that such kind of cancers, the epidermal growth factor receptor is a very attractive candidate for cancer therapy. The high level of receptor expression compared with the healthy tissue, the epidermal growth factor receptor plays a key role in the tumor cell and growth function. In the case of the lung carcinoma, it can if you now inhibit the epidermal growth factor receptor in these cancer cells, you can effectively inhibit all the downstream signaling, and therefore you can stop proliferation of the cancer cell. And if you now these such kind of inhibitors which inhibit the epidermal growth factor receptors in the cancer cells, they do not have very high levels of toxicity in the case of normal cells. So, similarly in the case of head and neck cancer, again the epidermal growth factor receptor inhibitors are being tried as a cure for head and neck cancer. Again, as you can see, the EGF receptor is overexpressed in almost 90% of the head and neck cancers, and this overexpression is associated with cancer and decreased survival, and increased EGF receptor expression is an early event in carcinogenesis and even present in pre-malignant lesion, that is even before the cells become malignant, you can see overexpression of epidermal growth factor receptors. So, if you can block, you can actually prevent the cancer becoming malignant. So, inhibition of the growth factor receptor tyrosine kinase slows the growth of the xenophage tumor models of head and neck. So, number of experiments have actually shown if you can inhibit the epidermal growth factor receptor signaling in these neck and head and neck cancer cells, you can prevent tumor growth. So, let us now try to understand what are the various strategies that are being devised to inhibit epidermal growth factor signaling. What kind of molecules are being designed? One approach is to design molecules which actually act as inhibitors of the receptor tyrosine kinase. As I said, the receptor tyrosine kinase to be active, it has to now bind to ATP. Now, if you can develop a decoy which will now go and bind to the ATP binding site of tyrosine kinase, therefore, the ATP molecule can no longer bind, and therefore, the receptor tyrosine kinase cannot be active, and therefore, one approach of treating cancer is to develop molecules which are very efficient inhibitors of the tyrosine kinase, and therefore, the tyrosine kinase can no longer be active, and therefore, genes involved in cell proliferation cannot be activated. Therefore, cell proliferation of cancer cells can be blocked. The other approach is to devise molecules like monoclonal antibodies which go and bind to the extracellular domain of the growth factor receptor. So, when these monoclonal antibodies go and bind to the extracellular domain, the epidermal growth factor cannot bind, and therefore, this signaling does not take place. So, only when the epidermal growth factor binds to the extracellular domain, it undergoes a conformational change so that it can dimerize. Whereas a monoclonal antibody binds, such kind of a conformation does not take place, therefore, the receptor cannot dimerize, and therefore, downstream signaling does not take place, and therefore, gene expression can be blocked. The other way is to devise antibodies which actually go and bind to the epidermal growth factor itself. So, when these antibodies go and bind to the epidermal growth factor itself, the growth factor cannot bind to the growth factor receptor, and therefore, the signaling is blocked, and therefore, you can stop the progression of the cancer cells. So, there are a number of such approaches. So, based on these kinds of approaches, a number of drug molecules have been developed, and you can see these are now available as gefitinib, erlotinib, cetuximab. Either these have all been approved for human use, or they are under various stages of clinical trials, and these molecules are highly selective and potent reversible EGF receptor tyrosine kinase inhibitors. So, they go and bind to the ATP binding domain of the tyrosine kinase, therefore, the enzyme activity is blocked. Cetuximab is actually a monoclonal antibody against the EGF receptor, therefore, the growth factor cannot bind, the EGF cannot bind, and therefore, the signaling is blocked. And there are also many other approaches, and so a number of approaches are being used to block the EGF receptor signaling, so that genes involved in cell proliferation cannot be activated, and therefore, cancer cell proliferation can be stopped. So, just like EGF, there are also a number of other growth factors which have been associated with cancer cells. Mutations or variations of these growth factors have been shown to be present in a number of other cancer cells and as a result all these growth factors are also targets for drug targets. For example, PDGF, original shown to regulate cell proliferation, also has a homology to a very important viral oncoprotein called VCIS. So this VCIS is actually has certain mutations in the PDGF, and as a result when this virus infects, the cell proliferation program is continuously activated and therefore it becomes cells become oncogenic and then it gets transformed and then leading to cancer. Similarly, there are certain cancer cells in which the TGF alpha is highly overexpressed, as a result it results in cancer. So basically this list of actually shows that a number of growth factor receptors have an oncogenic potentials and many oncogenes have been shown to encode variants of growth factor receptors. Okay. So I have just given some of these variants in the parentheses, and many of these growth factor receptors, either the EGF receptor family, the FGF family, or the PDGF receptor family, and so on and so forth, they all either variants or mutant forms of these receptors are expressed in cancer cells, or they are all present in the oncoviruses, and therefore they can result in cancer. So I think what I have discussed so far here is a very important signal transduction pathway, namely the growth factor and growth factor receptor signaling, and I have discussed very briefly how when a growth factor binds to a growth factor receptor, how the tyrosine kinase activity of receptor is activated, ultimately leading to the activation of a RAS, and RAS now results in the activation of MAP kinases, and MAP kinases now go and activate specific transcription factors which in turn activate genes involved in cell proliferation such as CFOS, leading to cell proliferation. And if this regulation of this pathway is lost, it results in uncontrolled cell proliferation leading to cancer. So what I have listed here is a number of important online resources that we can make use of. I myself have made use of many of these online resources. One can go to many of these websites and then look at these websites and see you can get a number of very important information on growth factor signaling. So one can just copy and paste any of these websites, web links I have given here, and then go to these websites and you get wonderful illustration of growth factor signaling. And many of the cartoons I have shown in this are also taken from some of these websites, basically to explain how growth factor signaling is very important. I have also listed a number of review articles here. One can go through these review articles and then get more knowledgeable about the growth factor signaling. There are also many reviews, there are original research articles, and last but not least I have actually listed here the two review articles written by the original discoverers of growth factors, namely the Stanley Cohen and Rita Olevi-Montalcini have written very nice reviews in JBC in 2008 and Scientific American, and one can go through this to understand how exactly growth factor signaling is very important. And finally I have listed the 3 landmark papers that have led to discovery of growth factors and growth factor receptors published in JBCs and Nature, namely the discovery of epidermal growth factor, the isolation of epidermal growth factor receptor, and the cloning of the human epidermal growth factor receptors. Some of these are landmark papers that actually change the entire growth factor signaling pathways. I think I will stop here.